All right, so for today and on Friday, we're going to be doing corrections on our problem set 1 for genetics and problem set 2. So we'll do problem set 1 today. What you're basically going to do is you're going to take out your copy of where you did your work, and then you're going to take a different color, whether it's like a physical pen or pencil, or on a PDF if you worked on the PDF directly. Take a different color, and you should be marking where you got it wrong, and then writing down the corrections based on what I'm telling you the answers are. If you actually got the entire question right and you showed all the work that I'm asking you to show, then you can go ahead and skip marking that question where your work is already shown. The important thing is to note that you can only get the extra credit points for these corrections if you already submitted your problem set, like I asked you to, before Wednesday. Because if I'm just giving you the answers and you just copy down the answers and you expect to get points from it, that's not fair to the people who actually did work. So it's not hurting you to see the answers, but I can't give you the extra credit points for it because you didn't earn the credit to begin with. Okay, so we're going to go through and I'm going to be switching between two pages. One of them I actually hand wrote out myself. So basically, when you go through a genetics problem for Punnett squares and stuff, you're going to want to be taking notes similar to the notes that I took down for the information the question prompt is giving you. And then that sets you up for success when you are actually coming to try to answer the question parts like A, B, C, D as the problem sets have. So for number 1, as you can see on the screen, it says pea plants grow to 2 heights, tall and dwarf. The tall trait is dominant to dwarf. That means it's going to be the capital letter, right? Like capital H and lowercase h be alleles for the height gene. And the questions are asking you what height is a plant with genotype HH, both capital. B, what is the genotype of a dwarf plant? C, what gametes could a plant with genotype capital H, lowercase h provide? And D, 2 heterozygous pea plants are crossed and 100 seeds are produced. How many tall plants are expected among the offspring? And then E, give the possible genotypes for the tall plants in part D. So in the problem set, for number 1, obviously you would first mark down that tall is capital H and dwarf is lowercase h, okay? And you wanna write out for yourself just to make sure that you have it in your references that tall is dominant to dwarf. So you know that that's the reason why it's the capital. Now, part A is asking you what the capital H genotype is, or the homozygous dominant genotype is. And that's a tall phenotype because it has at least one capital H. Part B is the dwarf phenotype. It's asking you what kind of genotype it has. And that would have to be the lowercase h genotype, or the homozygous recessive, because if there's even one capital H, it's automatically going to show a tall phenotype. Part C, they're asking what gametes a capital H, lowercase h heterozygous plant can produce, and gametes are going to be one of each of the alleles that it can give, right? So in this case, it has 2 versions of the same gene. One allele is the capital and one allele is the lowercase. So there's capital H or lowercase h are the possible gametes that this plant can produce. Part D is asking you for a cross between capital H, lowercase h heterozygous plants, 2 plants that have both the same genotype, and there's 100 seeds produced, which means that once you finish and you find the ratios, you still actually have to multiply by 100 to give actual individuals. You can't leave your answer as a ratio or a percentage. And this question, you must show a Punnett square in order to get points. If you don't show a Punnett square, you don't receive points, whether it's in here or it's in an FRQ inside of your actual AP Bio test. So you would show the Punnett square that is a cross between the capital H, lowercase h heterozygous plants. And so you're writing out the Punnett square, and then you show the ratio, and you multiply the ratio by the 100, right? So you would have 25 dwarf and 75 expected tall offspring. And then in part E, what would the possible genotypes for tall offspring be? Well, you can have capital H as a result here, or capital H, lowercase h heterozygous. Both of them will have a tall phenotype. So the possible genotypes would be both the homozygous dominant and the heterozygous. Okay, now number 2 is going to be about edible peas. So edible peas may be smooth or wrinkled. In breeding experiments, true breeding smooth-seeded pea plants and true breeding wrinkled-seeded pea plants make up the pea generation, the parental generation. The F1 or first filial generation is 748 smooth peas. The F2 generation is 2,519 wrinkled peas and 7,503 smooth peas. If you already just look at the ratio, it's kind of like 25% to 75% ratio, right? So which trait is the dominant trait is what A is asking. And if you're just making notes from the information they gave you in the prompt, well, true breeding, if you looked it up or if you already kind of guessed, means offspring always all look like the parent. So when you have a true breeding plant, it's usually a homozygous dominant, or if you have like a homozygous recessive cross, then obviously all the offspring are also going to look like the parents. So when you have a true breeding plant, the babies always look like the parents, and that's just literally what it means. So we know that the parental generation was smooth, crossed with wrinkled, and then the filial first generation is all smooth. There were 748 of them, and the guess is that they might be heterozygous. And is this possible to prove? Well, when you look at the second filial generation, there was about 75% smooth to 25% wrinkled, estimated ratio, right? It's not going to be perfect because like nature, things are just going to go awry, or it's just by chance, but it's really, really close, right? It's about a 3-to-1 ratio in phenotype. So yeah, the filial first generation is probably heterozygous. We're going to answer question A, and question A was saying, is the which is the dominant trait? And the easiest way to answer that would be which one has more in the second filial generation. And the second filial generation, the more or the larger group or larger ratio is the smooth side. So we're going to say that smooth is dominant. Part B is asking, is the F1 generation heterozygous or homozygous? And just by looking at the ratio of the filial F2 generation, you can already see and guess and make an educated guess that the first filial or F1 generation was probably heterozygous. And you have to write out the reasoning too, since this is asking for a sentence kind of answer. So F1 generation is heterozygous because the F1 cross produced offspring with different phenotypes. If all of the phenotypes looked like the F1 generation, then we would be assuming that the F1 generation had to be homozygous and true breeding. So part C is asking, approximately how many smooth peas in the F2 generation would be true breeding? And here's where you can kind of, you already know what the Punnett square looks like, because if the F1 generation are all heterozygous, the cross is similar to the cross right here. And you know that the capital H homozygous dominant is true breeding. And you also know that the homozygous recessive is dominant, is true breeding as well. So in order to kind of find the total and find the fourth ratios, you would just add them all together and then divide by 4. And that rounds up to 2,506. When you're rounding, you probably are rounding normally, like you round up or you round down based on the first decimal. And so we would be able to estimate around 2,506 true breeding are smooth, and then 2,506 are true breeding for the wrinkled phenotype. Now we're going to go to number 3. In mice, black fur, capital F, is dominant over brown fur, lowercase f. So you would be noting those two things down first because they gave you that information and you want to have it next to your work that you're about to do. In a breeding experiment, crossing a brown mouse with a black mouse produces all black offspring. Part A, what is the genotype of the brown furred parent? So we know black fur is capital F, we know brown fur is lowercase f, and we know that black is dominant over brown fur. So there's a brown furred parent and a black furred parent, and they produce all black offspring, which means that obviously we know that the brown furred parent has to be homozygous recessive, because if it has any capital F, it would automatically just have black fur. So we know that in part A, the brown furred parent has to be lowercase f, or homozygous recessive. Okay. In part B, it's asking what gametes are produced by the brown furred parent. Well, if the brown furred parent is homozygous recessive, then it can only give a lowercase f, right? No matter what it gives, it's going to be a lowercase f. It doesn't matter if it's the left one or the right one, it's going to be a lowercase f. Part C is asking, because all offspring are black, what must the black furred parent's genotype be, right? If there was like a 50-50 ratio of black to brown offspring, then you'd be able to reason that the black furred parent was heterozygous. But because all the offspring are black, that means that the black furred parent is only giving black furred alleles every single time, and it's dominant, so it's covering all the little f's, which means that the black furred parent must always give a capital F, so therefore it must be homozygous dominant. Part D is saying, what is the genotype of the black furred offspring? Well, if one of the parents can only give a lowercase f, then every single offspring is going to get a lowercase f. But since they're Blackbird, they have to have a capital F. So all of the offspring from this cross right here shown, this is your evidence, would be all Blackbird, but their genotypes would all be heterozygous. And then part E, if two F1 offspring are crossed, what is the phenotypic ratio that is expected? And the phenotypic ratio, well, we know the offspring are all capital F, lowercase f, heterozygous. So when you do that cross, you have to show this Punnett square as well in order to get credit. And after you show it, you would see the ratio would be 3 Blackbird to 1 Brownford, or 75 to 25 ratio. For number 4, the presence of horns in Hereford cattle is controlled by a single gene. The hornless or polled condition is capital H. Polled is just the name for it. And that's dominant over the horned condition, which is lowercase h. 15 genetically identical polled cows, aka they're capital, they have a capital H, are crossed with a single horned bull, which means that the bull has lowercase h, lowercase h. This results in 8 polled calves and 7 horned calves. Okay, this means that sometimes these cows are giving a lowercase h. They're not always giving a capital H, right? Because there's some calves that are lowercase h, and we know that the dad or the bull can only give a lowercase h. Okay, so you want to keep that in mind, and you want to jump to those conclusions immediately because they are valid conclusions to make. So what is the genotype of the polled cows? Well, we can just kind of answer that. If they're able to give a lowercase h for the to make babies that have horned, that are horned, then you know that they have to be heterozygous, right? So all the cows are all capital H, lowercase h. And it's kind of weird to say that, but the reason why they're all capital H, lowercase h is because it's given in the prompt that all of them are genetically identical for this gene. B is asking, what is the genotype of the horned bull? Well, that one's easy. We know that it has horns, so therefore it has to be lowercase h, because if they had a single capital H, then it would automatically be polled or non-horned. So that would be your answer to part B of number 4. Oh, good. My questions are not showing. Let me close it and reopen it. And we're going to be on number 5, where we're talking about fruit flies. Scroll down to number 5 and see if it will show. Perfect. Sorry, technical difficulties. Okay, in fruit flies, red eyes, capital E, are dominant over purple eyes, lowercase e. And this is actually real. Some of them have purpley eyes compared to the normal red eyes that you see. 2 red-eyed parents are crossed, and they produce an F1 generation of 76 red-eyed and 24 purple-eyed offspring. Hint, hint, that ratio is showing up again. A is asking, what is the approximate phenotypic ratio? So the approximate phenotypic ratio is basically using the number 76 and 24. So you would divide 76 by 24. And if you do that, then you would be getting around 3 to 1. And you need to write the ratio out with the actual phenotype. So it would be 3 red to 1 purple. Use my eyes if you want to. It's not as important, but you just need to get that down that it's about 3 to 1 for red eyes to purple eyes. Now, part B is asking, crosses between what 2 genotypes give this phenotypic ratio? And this is the traditional cross that we always talk about that gives 3 dominant phenotypes to 1 recessive phenotype. And that would essentially always be a heterozygous cross that we see, which means that the cross would look like this: capital E, lowercase e, crossed with capital E, lowercase e. Let's go over to part C now. Therefore, what must the parents' genotypes be? So if the parents give this kind of offspring ratio, then we know that the parents are going to have to be that ratio that we just predicted in B. So therefore, both parents are probably heterozygous because they have red eyes, but manage to produce offspring with purple eyes, right? Since they both have red eyes, that means they have a capital E, which is shown in the genotype that we predicted in B. In part D, what is the genotypic ratio for the F1 generation? Now, the genotypic ratio is different from the phenotypic ratio because in part A, we decided that the phenotypic ratio is 3 red to 1 purple. But we know that when you do a heterozygous cross, that if you show the work, you'll be able to see that there's a 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive ratio. And you have to show your work in a Punnett square in order to do this answer. Number 6, a botanist or somebody who studies plants knows that a certain cactus, that in a certain cactus, sharp spines are dominant to dull spines. So you want to write that down first. She is searching for a cactus resistant to herbivory, basically one that has sharp spines so that no animal is going to eat it. In her travels, this botanist discovers a beautiful sharp spined cactus. Does she know the genotype of this cactus? That's what they're asking in part A. So if you noted down that sharp is dominant to dull, then actually the answer is no. We know that sharp is dominant to dull, so the discovered cactus could either be homozygous dominant or heterozygous. We don't actually know its genotype because it would just have one dominant allele and it could show sharp spines, right? So we don't actually know yet. Maybe if you do a cross though, you'll be able to see. Part B, how could she determine the genotype of this cactus? Well, a test cross. So a test cross, if you look it up for yourself, or if you are learning it now, it's when you cross any organism with a known homozygous recessive. So if you cross them with a little lettered organism of the same species, then you are going to see if there are any homozygous recessive offspring that are produced. Because if there are, then you kind of figure out that the parent that you didn't really know the genotype of was heterozygous. And if all of the offspring are heterozygous and you look dominant, then you would also know that that parent that you were kind of about before, you would automatically know that those are homozygous dominant, right? Because none of their kids are homozygous recessive. So that will expose any recessive alleles held in the genotype of the organism of focus. So for this botanist, when she discovered this cactus, she goes, oh, it has a dominant phenotype, but I don't know if it's homozygous dominant or if it's heterozygous. If she doesn't test cross with a cactus that is dull spined or homozygous recessive, then she'll be able to see, are there any babies that are homozygous recessive? And if there are, you know that that parent that she didn't originally know is heterozygous. And part C is asking what kind of offspring would prove that her cactus is heterozygous. This is basically what I just explained. Sorry that I'm like over elaborating, but the more I talk about it, the more likely you'll be able to understand it. So a 50-50 split of dull to sharp offspring would mean her discovered cactus is heterozygous. So if that discovered cactus was heterozygous, the cross would look like this. And it's a test cross because you're crossing it with a lowercase. And if this was the parental genotype of the unknown cactus, then the results would be a 1-to-1 ratio of heterozygous to homozygous recessive. And it would be about a 1-to-1 ratio in phenotypes of sharp to dull. However, if the cactus that she discovered is homozygous dominant, the cross would look like this for a test cross. Remember, you're crossing with the homozygous recessive. And honestly, all of the offspring would probably end up being sharp spined. And that's because they would be heterozygous and they would always have a dominant allele. So the test cross is super useful in figuring out what the genotype of an unknown parent is. In number 7, petunia flowers may be red, white, or pink, right? We talked about this when we did the first note set with that first lab for genetics 1. Intermediate, the pink is the intermediate between red and white. So think about like that blending of color. Which one was that? This pattern of inheritance is called what, right? So the options would be codominance or incomplete dominance. And if you think back to your notes, then you would obviously know that the blending color is called incomplete dominance because when you have codominance, co means both show, co for both, right? Incomplete dominance, you get that weird kind of blending in the middle. But remember, the traits are still different. So if you keep reading the blended colored ones, you're still going to get different colors. You're not only just going to keep getting blended ones. Remember, they didn't like that term when I kind of described it to you, but it's an easy way to figure out that it is incomplete dominance when you see blending. So part B in 7 is, if a red petunia is crossed with a white petunia, what is the expected genotypic ratio? Well, you're going to write out the genotypes of the parents first. So we're just going to use C for color, right? But the color is going to be CR for red and CW for white, right? We're going to represent the parent separately because when it has like an R and a W, then it's going to be counted as a pink petunia. The reason why we're not just using R, capital R, and capital W is because if you use different letters, then you're representing different genes. So we need the C to represent the gene, but we're going to use the R and the W to represent different versions of the gene. Okay. So the cross is going to be CRCR with CWCW because it's a white petunia with a red petunia. And after you do that, you're going to see that all of the offspring are CRCW, which is 100% pink. Okay. So that would be the phenotypic ratio after you find the genotypic ratio, which is 100% CRCW. Part D says if the two of the F1 generation were crossed, what phenotypes appear in the F2 generation? So you're going to take those CRCWs and cross them together, and you must show a Punnett square again. So you're going to cross CRCW with CRCW, and you get this ratio of 1 red to 2 pink to 1 white. And it's kind of similar to the like dominant recessive one, except for the fact that the heterozygous ones do not look like the homozygous dominant ones in this case. Okay, and then in part E, it's asking you, what is that genotypic ratio? Well, the genotypic ratio would be the 25% CRCR. You can also do 1 CRCR, and then 50% CRCW or 2 CRCW to 25% or 1 CWCW, and that would be the genotypic ratio. For number 8, humans have curly hair, straight hair, or wavy hair, right? That's an intermediate, again, hint, intermediate between curly and straight. A wavy-haired woman and her wavy-haired husband bring twins home from the hospital. The son has straight hair, but the daughter has curly hair. A, has the hospital made a mistake? Oh no, we're gonna find out, okay? So when you write out the genotypes, you're gonna wanna, again, make note that the gene is represented by a base letter, but in this case, the base letter is not going to change. It's only the superscript that is going to change that represents the different versions of the gene or the alleles. So we're going to use H for hair because it's about hair, and we're going to use HC for curly and HS for straight. And we know that HCHS as a genotype is wavy hair because it's that intermediate. So we know that both of the parents have wavy hair. So they're both HCHS. So you're going to do that Punnett square to show the possible genotypes of their offspring and then the possible phenotypes of their offspring. So if you cross them, you get a ratio of 1 HCHC, which is curly, to 2 HCHS, which is wavy, 1 HSHS, which is straight. So we were talking about the son and the daughter, and the son had straight hair, and the daughter had curly hair, and that means that they have HSHS, and the daughter has HCHC. And if you look at the results of the cross, the answer to the question is no, the hospital did not make a mistake, right? Both children's phenotypes and their genotypes are possible from the parental cross, right? Because each time they have a kid, these are the chances, these are the percentage chances that they could be any of these physical traits. Okay, so that's what part B is kind of asking about. If these parents have more children, what is the chance that they could have a wavy-haired child? Now, the wording to answering this question is extremely crucial that you get this on point and exact like this. So the phrasing is, each time a child is born, there is a blank % chance that it has this genotype or this phenotype. And the reason why we have to phrase it this way is because you can't say that if a couple has 4 kids, they will exactly be 1 to 2 to 1 ratio for straight curly hair, wavy hair, and straight hair, because that's not true, right? The only thing we can produce with a Punnett square and predict with a Punnett square is the % chance that each time a kid is born, what is the possibility that they'll get this trait or this trait or this trait. Okay, so it's like a by chance basis. It's not like a overall, you should have this ratio. But a lot of the times, because the chances are based on the Punnett square ratios, if you have a lot of offspring, you might end up with that similar ratio, right? When you do plants and stuff. But people don't tend to have thousands and thousands of offspring from one set of parents, right? So we're not going to actually be able to see just by random chance the accumulation of enough offspring to see the ratio predicted by the Punnett squared. So we're going to go, each time a child is born, there's a 25% chance that child will have curly hair, a 50% chance that child will have wavy hair, and a 25% chance that they will have straight hair. So the question was focusing on if the child will have wavy hair, right? So that would be that the child would have a 50% chance of being wavy haired whenever a child is born. Okay, we're going to look at number 9. We're almost done. In humans, the L gene on chromosome 4 codes for a glycoprotein. Okay, so it's a protein that has a sugar attached to it. There are 2 functional gene products, glycoprotein M and glycoprotein N. there is no allele for gene L that does not result in a functional protein. Okay, there's no recessive. Okay, that's your key hint that this is not going to be normal dominant recessive already. Okay, a person with blood type M makes only glycoprotein M. A person with blood type N makes only glycoprotein N. And a person with blood type MN makes both glycoproteins. So this is an example where both are showing. And if both are showing, then this pattern of inheritance is called codominance. Okay. In part B, a man with blood type M has a child by a woman with blood type N. What is the man's genotype? Well, we know that if the man's genotype in this codominance situation and he has blood type M, that means that he has to have LM, LM. He has to have both M's, because if he had one N, then he would be blood type MN, right? If he's blood type just M, then you know that he has to have both M alleles for the blood type. And there's no recessives. That was the key to kind of figuring that out, okay? There's not going to be any like lowercase l because the gene is just capital L. Now we're going to look at part C. What is the woman's genotype? Well, we know that the woman has a blood type N, so that means she's going to have an LN genotype. Part D is asking, what is the child's genotype? Well, here you have to produce a Punnett square, okay? So you're going to do the cross between the man and the woman's genotypes, and you're going to do a Punnett square, and you're going to realize that all of them are LM, LN, which means 100% blood type MN, okay? And that would be your answer to the blood type of the child, right? The child, when they're born, can only be blood type MN. If there's anything else showing, they would be cheating, okay? And in our last question, in domestic cats, a cross between a tan cat and a black cat results in only tabby kittens, right? The tabby kitten is a combination of tan fur and black fur, which means both are showing, right? Not blended, both are showing. A cross between two tabby cats results in black kittens, tabby kittens, and tan kittens. First off, in part A, what is the genotypic ratio for kittens in a tabby-tabby cross? Well, you're going to actually have to do it, right? So we are going to use F, capital F for fur, and we're going to use capital FT for tan and capital FB for black for the fur colors, right? Because we know that this is a codominance case. We want to use the base gene, and then we're going to want to use different letters to represent the alleles. So the if we have a tan and blackford cross, then we would have 100% tabby offspring, right? And if we have a tabby-tabby cross, then we would be able to get all three as the prompt set, right? The prompt had tan kittens, tabby kittens, and blackford kittens as a result of this heterozygous cross. Now, part A is asking you what that ratio would be, and that's that 1 to 2 to 1 ratio of 1 tan, 2 tabby, and 1 blackford ratio. So you're looking at that F2 from an F1 cross. And honestly, at this point, I expect you to be able to show the Punnett square. So you want to show the Punnett square cross between the FTFB times the FTFB on the side here in order to produce this genotypic ratio. In part B, in a tabby-tabby cross, which kittens, black, tabby, or tan, are more likely? So you're going to look at that ratio we just produced, right? And which one has the highest number in front of it? That would be the tabby, right? Tabby FTFB is more likely because there's a 50% chance, and that's higher than a 25% chance of either black or tan. The last question is C. If you only wanted black kittens, what color parents would you choose? Well, it's pretty obvious. If you only wanted black kittens, you would only be able to guarantee that all the kittens are black if both the parents have black fur as well, which means both of the parents have to be FBFB. Okay, that's the only guarantee, and that would be your answer to that question. So again, as you're going through, pause at any point that you need to in order to correct your answers, or if you completely missed the question, feel free to kind of write down what I wrote down while following along with what I explained. And if you got it completely correct, then don't worry about marking on that question. So you're going to submit your corrections in the post below, and you will get extra credit points based on it. It's not going to be any amendment to your original grade. You're actually just going to get a bonus of up to possibly 20 points on corrections.